This story of young people coming to MIT and making a difference in the world continues. And that's really what we're about, is the older crowd who's moving off into the sunset, as David says, at some stage. And the pleasure is to introduce one of those who's a member of the Koch Institute here, Michael Hemann. Michael's an associate professor. He's been at MIT about 7 years or 8 years now, 8 years. He's established an international national program in the analysis of genes that cause cancer and then genes that cause resistance to therapy in cancer. He's an extraordinary young person, really has made major contributions both in research and in teaching, and he is a co-leader of our graduate program now. And we're so excited to have him. In addition to all these talents, as many people here are, he's a performer. He's a bass guitar player. And I'm part of Stand Up to Cancer, so I go to California and to Hollywood 1 or 2 times a year. It's totally lost on me. I mean, my wife, I come home and say, I met so-and-so and so-and-so, and she says, really? I said, who are they? But Mike is a performer. He's a rock star of molecular biology, and it's a pleasure to have you, Mike, to talk about your work.
Thank you for the kind introduction. I decided the tie wouldn't do any good. One of the, I think, the great things about standing on the shoulders of giants is that you rarely have to give talks face to face, so it's harrowing.
It worked perfectly in dress rehearsal.
It worked, yeah, exactly. So I'm going to talk a little bit about work done in my lab on a disease called acute lymphoblastic leukemia, so in this case B-cell acute lymphoblastic leukemia. You're going to hear from me talking about some research from our lab, and then you're going to hear from the Go Mitch Go Foundation, who are advocates and family members affected by the disease. So hopefully you'll get a perspective of some of the work done here, but also some of the rationale for the work done here. Logging on to the, okay. I think I'm mic'd over here. So some of the work is, as David described, relates back to some of the early attempts at treating patients with cancer. So as many of you saw, if you saw the Emperor of All Maladies, that really one of the first cancers that was worked on was B-cell leukemia. And a lot of work was done at the NCI by Emil Frey and Emil Freireich, which I think is one of the great name coincidences of all time. And if your name is Emil Frey and meet a guy named Emil Freireich, then you know that it's sort of karma. And they had this idea that you should construct regimens of combination cancer therapy. And their objective was not to think about mechanism, not to think about what's going to kill a cancer cell. Their objective was to give patients as much cancer therapy as possible without killing them. So they were much more concerned with toxicity than they were with on-target efficacy. So they picked drugs that had different off-target effects, some that had liver toxicity, some brain, some blood. And these combination therapies were, in the end, really quite amazing. So in the context of B-cell leukemia, up to 80 to 90% of all patients with the disease were cured. We had lasting remissions. Depending upon the kind of B-ALL, it would be as low as maybe 50%, but 20 to 50%, only 20 to 50% of patients were actually relapsing. And this is in an age when, thank you. these are Emil Frey and Emil Freyreich, who I think got their glasses at the same place as well. So again, they decided that they were going to assemble these drug regimens like CHOP. CHOP consists of 4 highly genotoxic or DNA-damaging chemotherapies and cell-damaging chemotherapies. But the outcome was really a terrific one. So this is a Kaplan-Meier curve, and essentially we're looking at survival over time, where again 80 to 90% of patients in some cases were being cured. Now the sad fact is this is still the reality. We give precisely the same therapies for patients that have B-cell leukemia. Our major criteria is giving patients as much drug as possible that they can tolerate, and we have essentially the same success rate. So what was really a huge advance in cancer therapy in the 50s and 60s, I think now represents a real frustration in our inability to improve upon these regimens. And that's particularly true when you realize that, of course, these 20 to 50% of patients that are failing are not percentage points there. People like Mitch Whitaker, who you'll hear about later, who represent a subset of patients that we can't cure using existing therapies. So why have we failed to improve treatment of this disease? Well, there are 2 major reasons. The first of which is that the current standard of care, this ability to cure 80% of patients, represents a real impediment to the introduction of new therapies into the clinic. These are obligate frontline therapies. You have to get CHOP therapy going into the clinic, and so it's very difficult to think or innovate around new therapies. And the second problem is, as David mentioned, these are really empirically derived therapies. They didn't really understand why they worked or didn't work, they just know that they work. And one of the problems with empirically derived therapies is it's hard to reverse engineer them. We don't understand why they work, and so we can't mess around with the component parts. And so the approach that we and other labs have taken to try to get at improving outcome in BALL is to try to model cancer therapy. And what modeling cancer therapy means that we want to generate tumors that look like the human disease or recapitulate the human disease. And we want to test therapies in an experimental setting prior to use in cancer therapy. So essentially, we need to build an airtight case that allows us to go to clinicians and say that we have a therapy that we know is going to improve upon this high standard of care. Of course, the problem with modeling cancers is that tumor genomes are really complex. This is a normal tumor cell, this is a cancer cell, a particularly I think astounding cancer cell that has this astronomical number of mutations and deletions and translocations. But needless to say, there are lots of alterations. And if we just look at 2 chromosomes, so copy number on 2 chromosomes in a cancer, here the dotted line represents a normal copy number, and everything above and below represents a change in copy number. So there are lots of different alterations. And so experimentally, how do we start reproducing these changes? How do we recapitulate these changes to try to model cancer biology and model therapy? Well, I think we have to look no further than the pioneers that are sitting in the front row. At least the way that I learned how to introduce cancer genes into stem cells and normal cells was using retroviruses, and this is a technique that was pioneered by David Baltimore. So we can add genes into cells to try to recapitulate the effect of overexpressing something. And similarly, by following what Andy Fire as a postdoc in Phil Sharp's lab did, we now have techniques where we can rapidly inactivate genes using RNAi, or more recent technology from MIT as well as elsewhere, CRISPR, which allows us to acutely delete genes in a genome. So effectively, we can introduce cancer genes into cells using viruses, and we can inactivate genes the same way. So right now, we really have a tool set that allows us to recapitulate cancer genomes. We can overexpress genes to recapitulate that state, and we can inactivate them. So I wanted to provide an example of how using this kind of biology can help us model cancer therapy. And this is work that was done by a postdoc in the lab, Christian Palash, and involves a kind of immunotherapy that David just mentioned. So in Christian's case, what he wanted to do is he wanted to essentially create a human tumor, a human B-cell leukemia in mice. And so he started with a virus that expressed human cancer genes, and he used it to infect human hematopoietic stem cells, the same ones you would use for a bone marrow transplant, and then transplanted these into mice, essentially humanizing them. So they have human hematopoietic cells in a mouse. And because we introduce cancer genes, we can create cancer in 100% of these recipients of oncogenes, here labeled GMB for MYC and BCL2. So we get tumors with 100% penetrance that are in fact human tumors. They bear human surface markers. They just tell us that it's a human disease. And it's a disease that we know is a BALL, and it's resistant to our frontline therapies. And so one of the things that Christian was interested in doing is, now that we have what's a human tumor, can we use human-specific therapies to try to target it? And the human-specific therapy he chose to use was an antibody therapy called Alemtuzumab. The specifics aren't important, but it's an antibody that binds a protein on a human cell. And using this as a single agent, he could see an effect. So in the red line were mice that received this Alemtuzumab, and the black line, those that didn't. Well, how does this work? Well, antibodies in this case work by binding target cells. So Alemtuzumab binds a leukemia cell, and then you have these functional members of the immune system like macrophages that find antibody-bound tumor cells and engulf them. Except in this case, it's not working all that well because all of these mice are relapsing on the disease. We're not getting a durable response. And so Christian looked at a variety of organ systems. So for example, he looked in the blood and the spleen and the liver, and we saw in each case is that the antibody therapy was able to clear tumor cells. So we're looking at a log scale, and we're getting 100 or 1,000-fold reduction in tumor burden in these organs. So it's effective in those organs, it's just not effective in bone marrow. So we have a site in the anatomy that we know is actually refractory to the therapy. And he wanted to know then, is the antibody actually making it to the target? And so you could look at this with a fluorescent antibody. So it gets to the spleen in this upper right quadrant. It doesn't get to the brain, as we know it doesn't cross the blood-brain barrier, but it does get to bone marrow. So it's getting there, it's just not killing target tumor cells when they're there. And so one of the great advantages of these models is that we can start looking at experimental combinations without experimenting on human patients. We can look at the addition of this therapy plus a variety of other therapies. And Christian did this looking at a variety of different compounds, a variety of different frontline therapies like radiation. And what he found is that there was a unique combination, a combination of cyclophosphamide, which is a frontline therapeutic in the treatment of ALL, but cyclophosphamide plus Alemtuzumab promoted this huge synergy. So you got about a 10,000-fold increase in the efficacy of the antibody when you just add this drug to it. And this resulted in really profound responses in our mouse models. And so here we have mice that get this combination therapy, and they actually live way out to over here. So they live until we actually remove them from our colony. We eradicate the tumor. Interestingly, this death here is actually caused by toxicity. It's not caused by the persistence of tumor cells, it's caused by drug-induced toxicity. And what's nice, again, with this kind of model is that we can deal with that. We can dial down the dose of cyclophosphamide and still get the same effect. So we can eliminate toxicity and we can eliminate tumor burden. How does this work? Well, it works by a curious mechanism. So it turns out that cyclophosphamide induces the release of factors from tumor cells that essentially make them more edible, right, by macrophages. And so an example is shown here where we can see green tumor cells being eaten by macrophages when we add cyclophosphamide. So we add this agent, we essentially vastly accelerate the ability of macrophages to do their job. They're just tumor coming up from the bottom that could get consumed. They can eat 4 and then they die. And so the model that he came up with is that typically the bone marrow is this repressive microenvironment. So it essentially inactivates the ability of macrophages to do their job. But you add cyclophosphamide at low dose, and it induces the release of these sort of eat me signals. These eat me signals then promote the ability of these local macrophages to then consume them. Now, the specifics of this story are not that important, but I think it does illustrate a few points. One is that we can use established drugs in new ways when we understand them, when we understand what they're doing, and actually use them in ways that allows us to dose reduce or pair with other effective chemotherapies. That these effective models of cancer therapy then allow us to build a case where we can go to clinicians and say we have therapeutic potential that is better than existing therapies and can be used at lower doses. I think most importantly, we can improve the standard of care for what we now call treatable cancers, cancers where we have 50 or 60 or 80% success rates, where we have 20 or 30 or 50% failure rates, that we don't need to be bound by the therapies that were currently used, that we can use, I think, innovation and modeling to change the standard of care in the clinic. And I'll end there by thanking all of you for coming, and also the Go Foundation, which you hear from in a minute, for their support as well. So thank you.